It's a good afternoon, and we know it's a morning to others in other parts of the world. Thank you to everyone for joining, and welcome to the HPTN 084 DSMB results webinar. My name is Ntando Yola. I am the Community Working Group co-chair for the HIV Prevention Trials Network. I co-chair the working group with Melissa Tenner. The HIV Prevention Trials Network is a worldwide collaborative clinical trials network that is dedicated to developing and testing the safety and efficacy of interventions designed to prevent the transmission of HIV. As part of this, the network recognizes the benefits of community participation in the research process, and this is in line with this commitment. It is the reason why we've put together this webinar. And it is with such delight that we'll be sharing with all of you the trial results of HPTN 084. And the results tell us that long-acting injectable cabotegravir as PrEP is highly effective in preventing HIV acquisition in women. And we will hear more and have an opportunity to engage on what these results mean. And we really hope that you will participate once we hear from all the presenters what these mean and where to from here. As a reminder to everyone on the call that please stay muted while our speakers are delivering their remarks. Please submit questions via the Q&A function in Zoom. And for all other remarks, you can use the chat box. I will now introduce our moderator, Michelle Warren, who is the Executive Director of AVEC. And he has been working with HPTN and other networks and other partners in advancing HIV prevention research. Over to you, Michelle.
Ntando, thank you so very much for convening this call. I want to thank you and Melissa both for the leadership of the community working group and for inviting AVEC and me to be part of this conversation. And want to welcome everyone to this call as well, and to say that we, together with the HPTN, with other networks, with many of you on the line, have had many calls over the years about HIV prevention, and they are always important conversations about what results mean and what to do next. But it is especially exciting when these conversations focus on such exciting positive news. It's been a remarkable week on many, many levels, and HPTN 084 results are a key part of that. And we don't often get to celebrate in the world of HIV, and certainly over these last many months with a new pandemic. So it is really exciting to see so many people on the line to share in this conversation. As Ntando said, we're going to have some initial presentations from the leadership of the trial, who I'll introduce in a minute. And then we will have an additional presentation from a great friend and colleague, Yolanda Matamba in Malawi, and then we'll have a conversation. As Ntando said, we really want to encourage that conversation, so please do put your questions and answers in on the Q&A feature as we go, and we will try to weave them all together in the latter half of the call for a conversation. So that's the run of show, and again, welcome all. I'm going to turn it over to two researchers who are also great doctors, who are also great advocates, and it's great when people can wear multiple hats. Sinead Delaney Moretle and Mina Hosseinapour have run HPTN 084 for a number of years, and you'll hear about the trial, its design, and its results. I think, as everybody on the line knows, these trials don't happen easily or overnight or with any one or two people, but it does take leadership, and Sinead and Mina have really been remarkable leaders of this program. Sinead is the Director of Research at the WITS Reproductive Health and HIV Institute, WITS RHI, based in Johannesburg. She has been really a pioneering researcher at the intersection of sexual and reproductive health, particularly amongst adolescents, and has worked on PrEP in many forms, as you'll hear in just a minute. In addition, Mina is based in Malawi. She's the scientific director of the University of North Carolina Project Malawi and has been based in Malawi for almost 20 years now, where she's an infectious disease physician and a leading prevention researcher. So without further ado, I'm going to turn it over. I think, Sinead, you're going to start with some slides, and I'll turn it over to you and Mina.
Thanks very much, Mitchell, and good afternoon to everyone. I'm going to share my screen. So thank you very much, everyone, for taking your time to attend this call. We have important news to share about the HPTN 084 study. In today, we'll review very briefly the HIV Prevention Trials Network to those of you who are unfamiliar with it, talk about the HPTN 084 study design, share with you the preliminary results and next steps, and then, as Mitchell said, we'll have the opportunity for a conversation. So for those of you who are unfamiliar with the HPTN or HIV Prevention Trials Network, as Ntando mentioned, the HPTN is a worldwide collaborative clinical trials network that brings together investigators, ethicists, community, and other partners to develop and test the safety and efficacy of interventions designed to prevent the acquisition and transmission of HIV. HPTN studies evaluate new HIV prevention interventions and strategies in populations and geographies that bear a disproportionate burden of HIV infection. The HPTN has more than 150 sites in more than 15 countries. And today we'll be speaking to you about the results of one of the research projects in the diverse portfolio, HPTN 084. So first, I'll remind you of the study design of HPTN 084. HPTN 084 is a phase 3 multi-center randomized double-blind controlled trial that was designed to evaluate the efficacy and safety of long-acting injectable cabotegravir to prevent the sexual acquisition of HIV in cisgender women at risk of HIV infection. The study compared the effectiveness of cabotegravir to daily oral Truvada. Truvada has been shown to be highly effective for HIV prevention when taken as prescribed in a variety of populations and contexts. In HPTN 084, study participants were randomized in a 1-to-1 ratio to one of two study arms. One arm received active cabotegravir and a Truvada placebo. The other arm received active Truvada and placebos for cabotegravir. Cabotegravir was administered daily by mouth for 5 weeks and then via intramuscular injection at 8-week intervals after an initial 4-week interval load. Participants in both arms were offered open-label daily oral Truvada for 48 weeks after their final study injection. And here you can see that study design laid out. I'll now review the study population that was enrolled. A total of 3,224 HIV uninfected participants were enrolled between November the 27th, 2017 and November the 6th, 2020 at 20 sites in 7 countries in sub-Saharan Africa, including Botswana, Eswatini, Kenya, Malawi, South Africa, Uganda, and Zimbabwe. A total of 3,127 participants were included in the analysis at the time of the DSMB review. The mean age of study participants was 26 years old, and 57% of participants were 25 years or younger. 82% of the women enrolled were not living with their primary sexual partner. 55% reported 2 or more partners in the past month, while 34% reported a primary partner who was either living with HIV or of unknown HIV status. Recognizing the challenges of COVID-19, the HPTN 084 study team developed plans responding to the impact of the pandemic on study conduct. The plans included significant measures to protect study participants and site staff during COVID-19. The team also proactively established metrics of disruption and used these to assess whether an administrative censoring of data would be needed during potential periods of disruption. I'm pleased to tell you that at no time during implementation of the study did we meet the pre-specified disruption threshold for censoring of data. As of November the 6th, 2020, participant visit completion at months 6, 12, 18, and 24 was 94%, 91%, 89%, and 91% of expected. I will now tell you more about the NIH Multidisciplinary DSMB review. The Data Safety and Monitoring Board is an independent group of experts that advises the HPTN 084 protocol team. The members of the DSMB have primary responsibilities to review and evaluate the study data for participant safety, conduct, and progress, as well as efficacy, and to make recommendations concerning the continuation, modification, or termination of HPTN 084. I am pleased to inform you that the blinded portion of the study reached its objective, and the DSMB recommended stopping of the blinded portion of the study after its review on November the 5th, 2020. The DSMB considered the analysis of the preliminary findings of 38 incident cases of HIV infection. These 38 infections are included in the intent to treat analysis and are all infections deemed to have occurred after enrollment and are included in the analysis regardless of whether participants continue taking their assigned study drug. The overall HIV incidence amongst all study participants was 1%, suggesting that both CAB and Truvada are highly effective for HIV prevention in this population. 34 incident infections were detected in the participants assigned to the Truvada arm, equivalent to an incidence of 1.79%, and 4 incident infections were detected in the participants assigned to the cabotegravir arm, equivalent to an incidence of 0.21%. That is, we observed roughly 9 times the number of incident HIV infections in the Truvada arm compared to the cabotegravir arm. The hazard ratio in the cabotegravir versus Truvada arms was 0.11, with a confidence interval of 0.04 to 0.32. The interpretation of this finding is that cabotegravir is superior to Truvada in preventing HIV infection in cisgender women, and the threshold for early stopping of the blinded portion of the study was crossed. Based on these efficacy findings, the independent DSMB recommended that the blinded portion of the study be stopped early and results released to the broader scientific and stakeholder communities. In addition, in a random sample of approximately 362 participants receiving Truvada, plasma tenofovir concentrations were measured. 64% of these samples contained detectable concentrations of tenofovir. 48% of specimens had concentrations of tenofovir consistent with daily use. This suggests that the HPTN 084 study population was more adherent to daily Truvada than had originally been anticipated, likely conferring an even higher level of protection against HIV infection for Truvada recipients than had been assumed in the original trial design. Currently, concentrations of tenofovir in plasma and tenofovir diphosphate in red blood cells for all those who acquired HIV infection after being assigned to the Truvada group are being evaluated. In addition, concentrations of cabotegravir in plasma for a subset of participants and for all those who acquired HIV infection after being assigned to cabotegravir are being evaluated. An analysis of the timing of incident infections in participants receiving cabotegravir relative to their last known clinic-provided injection are also underway, and we are also conducting resistance testing for incident HIV infections. I will now hand over to my co-chair, Dr. Mina Husseinipour, who will share the safety results.
Thank you very much, Sinead. From a safety perspective, both cabotegravir and Truvada were safe and well tolerated in HPTN 084. Most adverse events were mild or moderate and were well balanced between the arms. GI symptoms were slightly more common on the Truvada arm, particularly nausea. Injection site reactions were more commonly observed in the cabotegravir arm than in the Truvada arm. Injection site reactions were reported by 9% of participants in the Truvada arm and 32% of participants in the cabotegravir arm. Injection site reactions were most commonly mild to moderate pain and or tenderness and were mostly experienced at the first injection. 0 participants in either arm discontinued injections due to injection-related adverse events. I'm now going to review the immediate next steps for the HPTN 084 protocol. All institutional review boards, ethics committees overseeing this study, site investigators, and the study participants are being notified of these results as soon as possible. Participants will be informed of the medication they received. A protocol amendment will be submitted for regulatory review to allow participants to continue taking their assigned medication or to switch to cabotegravir long-acting injectable if they choose to. Participants on Truvada will be offered open-label cabotegravir as soon as the medication can be made available. The oral lead-in will be optional for participants who choose to switch from Truvada to cabotegravir. All participants currently on the study will continue to be followed on the study. Participants choosing not to remain in the study will be referred for the best locally available HIV prevention services. We are in the process of making these results available to national drug regulatory authorities. We look forward to presenting these results in peer-reviewed settings at upcoming conferences and in manuscript form as we continue to finalize primary analysis and begin work on multiple important secondary analyses. I am now going to provide some information on other studies involving cabotegravir. Earlier this year, a sibling study entitled HPTN 083 in cisgender men and transgender women showed a PrEP regimen containing long-acting cabotegravir injected once every 8 weeks was superior to daily oral Truvada for HIV prevention among cisgender men and transgender women who have sex with men. Open-label adolescent bridging studies are poised to provide critical safety and acceptability data in adolescent and young adult populations in support of regulatory approvals for youth. And sub-studies in HPTN 084, including a contraceptive sub-study, a qualitative sub-study, and a sub-study of women who became pregnant during the trial, will continue as planned. Let me hand back to Sinead.
Thank you, Mina. In conclusion, we'd like to take this opportunity to congratulate all the participants, their communities, the community advisory boards, the investigators, and the site staff on this tremendously exciting result. This great success is a shared success for all of us, and one we hope will lead to expansion of HIV prevention options for at-risk cisgender women globally, and ultimately reductions or elimination of HIV acquisition. We recognize that we are here because of the tremendous collaboration of many stakeholders. On behalf of my co-chair, Dr. Husseinipour, we appreciate the support of Scott Rose, Jennifer Farrier, and our colleagues at FHI 360, the Statistics and Data Management Center, the Laboratory Center, the HPTN Network and Network Leadership, and particularly the HPTN 083 team who went before us, as well as our in-country partners. We wish to acknowledge the study sponsor, the Division of AIDS at the National Institute for Allergy and Infectious Disease, or NIAID, and the funding partnership of NIAID, the National Institute for Mental Health, ViiV, the Bill and Melinda Gates Foundation, as well as the generous contribution of the study drug by Gilead. And mostly, I think we want to thank all of you for your support, particularly during these unprecedented times of the COVID-19 pandemic. We look forward to further conversations about the HPTN 084 results. Thank you.
Great. Sinead and Mina, thank you both so very much. I know at least some questions are beginning to come in, and I want to encourage people to continue to put questions in the Q&A feature, and we will get to them in just a minute. But before we do, I'm delighted to welcome Yolanda Matamba, a great advocate from Malawi, who is the Malawi country director for advancing girls' education in Africa, who's worked as a tireless advocate for HIV prevention and sexual and reproductive health for young women for many years, including time as a fellow in our AVAC Advocacy Fellows Program. So I'm delighted to welcome Yolanda to provide some perspective on what these results begin to mean just in this first week of of knowing about them and potentially their implications. So Yolanda, over to you.
Thank you very much, Mitchell, and to all the panelists. Good morning, good afternoon, and probably good evening, depending on which side you are this time. As I've already been introduced, that I'm the advocate for HIV prevention, specifically targeting for adolescent girls and young women, and also working tirelessly for women health. We are delighted this afternoon, basically because of the great news that we all know. But before I present the perspective of the great news, let me just give a quick background of which everyone else in this room today, we are all aware that HIV has really hit it so hard, adolescent girls and young women, which accounts for almost 50% of the new incidences, and precisely more happening in the Southern Africa, where also these studies were conducted. So as adolescent girls and young women, they've been facing a lot of challenges, precisely which put them at risk of contracting HIV. And we also know that options have not been, so many of them in the prevention basket that would be able to help women and adolescent girls to prevent HIV. So with more incidences happening in terms of sexual exploitation, sexual abuse, rape, as well as living in discordant relationship, has all put women at risk, and also without much protection. So the great news this afternoon of HPTN 084 has really brought some excitement, and we are all delighted that the CAB-LA results has been presented as early as we anticipated. And for us as women, and also representing adolescent girls and young women, we feel like this is a great opportunity and the greatest milestone, as we are looking forward to have more options in the prevention basket, and also as we are fighting together to respond to the pandemic. So from the southern part of Africa, and indeed in Malawi, say that the great news of CAB-LA will help more women to access the prevention that is discreet and long-term, and also something that for those that cannot be able to manage a pill every day will give an opportunity, because as women do have different needs at different stage of time, depending also on what is their preference. So as much as we know that we have options that are daily, that you can administer every day, but also we are mindful that depending on the different needs of women, we do need to provide different options. And the CAB-LA just bring that option to the prevention basket. So as an advocate, I would say that this news really has brought some kind of excitement, and would be looking forward to the regulators and all the authorities that the product should be able to reach those people that need the most in the very nearest time. We know the timelines that are already has been set to get the approvals from the FDA and all that, but the pre is to ensure that we all move together so that the option should be able to be available at the very nearest time in the southern part of Africa, because that's where the pandemic has really hit it so bad. And if we need to reduce the infection rate or the incidences, we really need to ensure that we move very fast with the options that are really very supportive, have been approved, and especially this one, the long-acting. I would say that we are so excited and we are grateful. And also, I would want to take this opportunity to thank all the study participants, especially the women that have been taking much time tirelessly to participate in the studies. I think it shows more commitment in the HIV prevention, where women in the southern part of Africa and indeed globally, we are involved in all these studies to ensure that the options that manage the needs for women are readily available, and also that can be able to be used anytime they need, whether it's a daily dose, monthly, or the long-acting prevention option. Thank you.
Great. Yolanda, thank you so very much for that perspective. And I think that the emphasis on options and choices is something that I know everybody on the line thinks a lot about. And thank you so much for giving voice to that. I'm going to try now to navigate us all through questions. A lot came in the Q&A, which is great. A few came in on the chat, which I'll also try to get to. I want to encourage people to use the Q&A, but I'm going to do my best to get us to all of it. Let me start, I think perhaps Sinead and Mina, a really important question from Patricia Jaconia about age. I know in other trials, we've often seen differentials in ages. So a question here first about, do we have data about younger women, 18 to 20 or 21? And were there any differences in efficacy based on age? Obviously, it's still very early and the numbers are small, but let me start there.
Thanks, Mitchell, and thanks to Patricia for that question. The trial involved women 18 to 45 years of age, but you heard that about, well, the median age was 26 and 57% of the population were under the age of 25. So I think in response to the question about 18 to 20-year-olds, we could estimate that probably a quarter of that number were between 18 and 20 years old. As for whether there are differences in efficacy by age, those analyses are still ongoing, so we're not able to say that yet. But we really recognize the importance of getting additional safety and acceptability data in younger populations. And that is why the HPTN 084-1 study is ongoing. And maybe to preempt the question that was asked in the chat about whether it's still ethical. So that study is an open label bridging study. It's not blinded. Participants will be enrolled and will receive open label cabotegravir. And given these results, we think there will be interest from young women to join that study in order to generate additional safety and acceptability data.
Great. Mina, you mentioned some of the side effect issues, and actually a colleague of mine, Navita Jain, asked about the difference in injection site reactions. I think you reported for the CAB arm about a 30% injection site. If I recall, 083 was closer to 80, if I'm not mistaken. So a curiosity there about if you have hypotheses about differences in reaction.
Well, women give birth, so I think that we have higher pain tolerance. So that was supposed to be somewhat funny, but
true.
I think that women are used to having injections through family planning, and so may not have perceived the effect of having the injection as something that was particularly painful or resulting in tenderness. So it may be a familiarity with having injections regularly, and perhaps fat content or something related to different body habitus. They're both IM injections, so I'm not sure if the body is an issue, but those are, I guess, some of my hypotheses. Sinead can add, perhaps.
I think Mina's right. That's what we've speculated, that women may be more used to receiving injections, but also the sort of increased adipose may have minimized some of the discomfort associated with an IM injection.
Super helpful. And actually, related question from Mark Hubbard. Were most of the injection sites at the first dose, do you see people kind of becoming more comfortable or more familiar with injections over time?
Yeah, I think this was a finding that we'd even observed in 083, but also in 077, which was the safety and acceptability study. And it does seem that people become habituated to the discomfort associated with the injections.
Great. A really important finding you reported that I think sometimes gets lost, and actually Jim Pickett highlights it, about what we now know from at least 084 about oral TDF-FTC. And I know you both have had experience with oral PrEP, and Sinead, I know many programs are reported that, oh, women in Africa aren't taking oral PrEP. You all seem to have found a very different reality there. And I wonder if you both could help us, and Yolanda too, what's the message about oral PrEP coming out of 084?
Yeah, Mitchell, I think that's a really important point, and it's one that we've tried to emphasize because I don't think we necessarily want to pit prevention products against each other. I think it's an important message that overall in this trial we saw an incidence of 1%, and that's pretty impressive when you consider some of the other HIV prevention trial results we've seen recently. What we just saw was that cabotegravir was much better at preventing HIV infection, but even in the Truvada arm, incidence was 1.79%, so under 2%, and sort of consistent with what we've seen in terms of the preventive benefits of Truvada. And I think it's a really important message to communicate. And maybe I think to get to Jim's question about sort of how effective is Truvada, obviously, this was a comparison of two active products, but one of the things we have worked on are counterfactuals, and I'm sure that with time and additional analyses, we will be able to estimate what this might have meant sort of in terms of protection in the absence of any intervention.
Super helpful. I don't know, Yolanda or Mina, if you want to add anything to that?
Yeah, I think what Sinead just presented is right, that the emphasis should be that they are all effective and that the drugs that works, and wouldn't want to place the other drug to be more superior than the other because they are all helping, but the emphasis should be about options because others might prefer the pill and others might prefer the oral PrEP and others Cap LA. So the most important aspect or the message to send across is that there are all these drugs that work effectively, and women can be able to choose which option is much, much better for them. Those that cannot be able to adhere to the pill should be able to take the vaccine, and those that are not very comfortable with the vaccine, then they can leave the jab and be able to get the pill.
If I might add as well, I think that as we get more data on the levels of the drugs and our additional analyses, we'll be able to perhaps characterize the women that struggled more with adherence to the oral pill and perhaps be able to, figure out how to assist those women to take the pills, or they may be the people that we want to promote the injectable to.
Mina, that's so helpful, and it actually leads into some questions that relate to the support that the trial provided. And I think Sri Gulab asked a great question about, can you describe a little bit more about any of the behavioral support that might have been provided, the adherence support, because you did see such a higher level of adherence? And as you think about answering that, there was another question, and I can't remember who asked it, but about were all of the participants in 084 what we would call PrEP naive? Had any of them had experience with oral PrEP before entering 084? And so that's one part of it. And then can you describe a bit about the behavioral social adherence support you might have provided?
So Mitchell, maybe I can respond and Mina can add. Our goal was to enroll women who were interested in PrEP, but I think at the time when we started in 2017, PrEP programs in the region were nascent, so probably we recruited a large number of women who in fact had not had PrEP, and this was one of the lessons that we learned early on, and that I think the sites did a tremendous effort on, was we experienced a period where participants would start on the trial but then discontinue because they would receive pushback from family and friends about taking pills, and an enormous effort was undertaken actually to educate communities about PrEP, and we I think were able to stop those discontinuations. So I think kind of we did this trial in an environment where very few people had experienced PrEP, and I think kind of the sites did excellent work to communicate the value of PrEP for HIV prevention. Mina, I don't know if you want to comment on the second part of the question about adherence support.
I think each site really generally provided a very supportive environment for their participants. whether it was through engaging their participants in group activities or just one-on-one support with the staff at the sites. So certainly they had a good understanding and education about PrEP and its adherence importance, and that may have been more than in what has been shown in demonstration projects.
Yeah, really helpful, and I think it may be really important for the trial. I know the PTN will do this. There may be some really great insights about some of the work happening at the site level that could be popularized into PrEP programs outside of clinical trials. So just I want to put a pin in that one. Sinead, it looks like you might want to add to something there.
And well, Mitchell, I think sort of one of the things we were aware of, because I know, kind of at the time, HPTN 082, for example, was looking at drug level feedback, but the the adherence support package that we focused on, I think, was a very good support package, but it was aimed at sort of being benchmarked with what the current national programs were delivering in order to kind of reflect sort of the practice in the communities in which people were living. So we didn't have bells and whistles in the sense of drug level feedback or additional measures. And so I think, again, it's a testament to the participants and their desire for effective prevention products, that they appreciated that they could be on an active product and took their pills as well as they did.
Great. Now, one of the things that Yolanda mentioned about options and choices, of course, in the clinical trial, women were randomized to the two arms, but I can't remember who asked it, so I won't be able to name you in the Q&A. I apologize, but there was a question about how long now this open label period will go. And I know, Mina, you talked about it, but and again, I think like we heard with 083, there's a period of choice that people will have between the two products now, and then the formal open label extension. And a question was, how long will that last going forward? So I wonder if you can maybe look at the next couple of steps again.
Yeah, I think it's a great question. The next step is to communicate this information to participants and to unblind them to their treatment allocation. There'll be some work on the part of the study team to organize study medication, to sort of alter the trial procedures, and then participants will be given an option to switch to open label cabotegravir if they wish. And I think it's going to be important to learn more about people's choices for prevention options in this context. With regard to length, a couple of things. Essentially, it's being negotiated, but what I would say is that the timeframes for this study, we originally anticipated it would end in 2022. So I think that gives us a kind of a bracket around which to work. And I think we were really sensitive to the issues around post-trial access, and have certainly heard from a number of sort of regional stakeholders the importance of this given the result. And so those are both important considerations for thinking about the length of the open label extension. But certainly we wish to retain as many people in follow-up as possible.
Great. And we're going to get to some of the next steps about the product. And I know there's some questions there. I did want to come back to one really quickly, just because it goes back to something you talked about before. Moses asked a question about, Moses Batanganya, about if you have knowledge of their women's contraceptive use, and if that might have influenced their reactions. So if there were many oral contraceptive users, I think it might be also helpful to maybe describe the contraceptive study that, Mina, you described, just to give people a little bit more insight into what we're hoping to see out of that study.
Mina, do you want to respond?
Sure. In the contraceptive sub-study, we're looking at the drug interactions between the implant, Depo-Provera, and NET-N to see if there's any effect on contraceptive efficacy.
And I think maybe just to add to, or to sort of respond to the question, it might be important to remind people that because of the concerns around periconception dolutegravir that I think emerged in mid-2018, we had very strict requirements for long-acting reversible contraceptive use in the trial. So we had about 2/3 of participants on an injectable contraceptive. So we can certainly do that analysis to look at correlations between ISR and contraceptive use, but also to recognize that we had a somewhat distorted contraceptive population.
That's really important point in all of that. Two questions kind of added on to this issue of the choice phase, and I want to just make sure we have clarity. Sinead, you mentioned that women on the oral arm would, as the unblinding happens, would be offered the opportunity to go on to cabotegravir. Jim Pickett asks, at the same time, wanting to confirm, and as with other trials, that women on they can stay on oral TDF FTC, or can women and can participants who are on the injectable, active injectable arm, can they switch if they choose? What are the pathways for that? And I think a question similarly from Jessica Larici, will you be collecting that data and reporting out? So a chance to really learn about that choice.
Yeah, I mean, just to say that once participants are unblinded, they'll be offered the opportunity to stay on their existing method or to switch. So Truvada to cabotegravir, but also there may be some people who are on cabotegravir who wish to stop injections. The one important thing for us to do in that instance would be to assess their ongoing HIV risk, and if necessary, to offer them additional open label TDF FTC to cover the PK tail.
Great. And I'm so glad you said the word tail. Like a conversation about long-acting injectable prevention would never be complete without talking about both the run-in and the tail. I know there were questions from a couple of actually dear friends and colleagues about this issue. Maybe we could take those in pieces. So on the oral run-in, you described, Sinead, in the trial design that you had this run-in of oral cabotegravir for those on the cabotegravir arm. And I think, Mina, you described that would now be optional in the next phase. And I think several people asked about on what basis does that become optional, and was there specific safety data? And so maybe if you can answer that, and then I'll shut up and let you answer, but maybe after that, if you could answer about this so-called tail. Obviously the trial is still ongoing, and how much information, if any, you have about what is needed for covering that pharmacologic tail.
Yeah, so I can respond to the optional oral lead-in. The recommendation about the optional oral lead-in comes from our colleagues at ViiV, and they may want to comment more after review across sort of the entire program. Just to remind everyone, the idea of the oral lead-in was in order to sort of exclude the possibility of any sort of tolerability issues prior to administering a long-acting injection, because once that's administered, it's difficult to reverse. I think Mina can agree, in our own experience, and without sort of having the numbers yet to hand, we didn't see any significant reactions that kind of were of concern to us. So we think that it's reasonable to now offer people the option of the oral lead-in. And obviously that's also based on data across the program. And then to respond to your question about the tail, and yes, I was giving you an opportunity because I've seen that question. Kind of it's true in the trial, less than 4% of women actually discontinued injections. And so we have a very small amount of information about people once they discontinued injections and that tail phase. And this is why we think it's really important that participants, all participants, irrespective of their product choice, actually are followed up during the open label extension so that we can learn more information about the tail.
Great. You did mention that some of that come from data about cabotegravir, the program writ large that ViiV leads, and delighted that both Kimberly Smith and Alex Reinhardt from ViiV are on the line as well. I don't know, Alex or Kim, if you have anything you want to add about the lead-in and the tail from the larger program, but also, while you're on, and delighted to see you on camera, Kim, there are obviously questions. As great as this data appears right now, the key question, I think, and Yolanda nailed it at the very beginning, when, what's next? And as someone asked, I can't remember who it was in the Q&A, when can we expect access in East and Southern Africa?
Thank you, Mitchell. Great questions. Let me first start by actually, on behalf of ViiV, I want to thank Mina and Sinead and the study team, as well as all of the sites and all the study participants for their contribution to this incredible study. Obviously, this was a unique partnership between ViiV Healthcare, Bill and Melinda Gates Foundation with the NIH, both DAIDS and NIMH. And we also like to thank our colleagues at Gilead for the contribution of Truvada for this study. So we're thrilled with the results, obviously. So let's answer the questions that have been raised. The first one is when will it be available? And I wish I could answer that one specifically, but let me just say that we are working with regulatory agencies in the US. We will also be working with agencies in South Africa and other sub-Saharan African countries and Europe to work through how we can get the process for getting the product approved as soon as we possibly can. And that is a high priority. The next question is, will there be access? And I can say 100% that ViiV is committed to access. We're committed to access to all of our products. The best example of that is obviously dolutegravir, where there are more than 8 million people on dolutegravir around the world. And that's because of ViiV contributions and as well as our partnerships with generic companies through royalty-free licenses and voluntary licenses in order for them to be able to produce the drug at costs that allow them to be available across the world. The same type of process is what we would intend for cabotegravir and all of our products. And so we are committed to making it accessible to people, to these young women in sub-Saharan Africa. And we certainly will work with the health ministries to do everything that we can to make that possible. So then the question that you asked about the oral lead-in. So we are developing cabotegravir for both treatment and prevention. In our treatment program, we have a study where we have allowed individuals to have the oral lead-in be optional. And when we looked at that, we found that really there was ultimately no difference in the PK or in the outcomes in individuals who had used the oral lead-in or not. It was for the purpose of identifying any potential safety issue, most importantly, a hypersensitivity reaction, which we have not seen throughout the program. And obviously there are many thousands of individuals that have been exposed to cabotegravir now. And so for our treatment program, we are looking to eventually make it an optional oral lead-in. And so for the prevention program, we really think that it's quite reasonable to get additional information about going without the oral lead-in in this clinical trial. And so that is that's the intent for the oral, for the open label extension. And then there was also a question about post-trial access. Certainly we're committed to making the drug accessible to the women who participated in the trial until it is approved and available in their region. And then the last question, I think that's the last question about the tail, and that is, so we have a bit of information, particularly from the 077 study that tells us that the tail lasts a year or more. And I think as Sinead pointed out, really the decision about whether or not the tail needs to be covered when we get to the real world will depend on the individual's overall risk. In the clinical trial, there is an option for providing oral Truvada to cover the tail for women that discontinue cabotegravir as a part of the trial. I think I answered all the questions.
Great. No, Kim, you did. I think you touched them all. And clearly there is lots to come in all of these conversations. I'm conscious of time, and there are some additional questions that I wanted to get to. That I might just jump around a bit because I think there's some really interesting ones. Chris Acola asked a question about any knowledge of the ART status of the HIV positive partners I think getting to this idea of work, Mina, that you and Mike and many others did, U equals U. Do you have any sense of partners' treatment status and if that might have had any implication on incidence?
We haven't looked at that data specifically, and what the question we asked at baseline was more whether they knew their partner was positive or unknown status. So we do have that information available that we'll be able to evaluate, but specifically whether their partner was suppressed, I don't think that we'll have that.
Okay. Very helpful. There was also a question, I think we'll all spend a lot of time comparing 083 to 084. We talked a bit about the side effect profile. Someone asked if, why we might have seen higher efficacy in 084. And in terms of the comparison between oral and injectable, the superiority margin was better, higher in 084. I suspect we all have hypotheses. Sinead, Mina, anything you want to share on that one?
Yeah, I think it's important for people to remember that the trials were designed slightly differently to begin with, but it's been interesting comparing the results to see some sort of consistent messages. I think in this trial, the reason that we saw such a significant difference between injections and daily oral pills is that the injections overcome many of the barriers that women experience with daily pill taking. And we had always suspected that in women, we may have less forgiveness around pill taking than we do, particularly in cisgender men or trans women. So it's probably a combination. When we looked at the adherence levels in women, we saw that I think it was 64% had detectable levels of tenofovir. So reasonable adherence, but we did notice that that did decline with time. And it's likely that those sort of barriers to daily pill taking may have been where cabotegravir was able to achieve an advantage.
Interesting. And while we're on that, I wonder, and I think we talked about this earlier in the week, any differences between sites and countries with respect, particularly to oral PrEP, because as you said, at the start of the trial, PrEP access was nascent throughout the region, but it is quite different in the different countries in which you conducted the trial. Did you see any variation?
Variation in?
I'm sorry, variation in oral PrEP adherence. Maybe thinking that countries with more advanced PrEP programs provide a better backdrop for PrEP adherence.
Yeah, there certainly were differences by countries, and there was definitely a range of adherence from about 45% through to 77%. And your hypothesis that this may be related to sort of familiarity with the products makes absolute sense, and certainly argues for sort of strengthening our PrEP programs in the region.
Absolutely, it'll make it much easier to introduce both dapivirine vaginal ring in the new year, hopefully, and cabotegravir the year after, we would hope. I know we're coming toward the end, and I want to give all three of you a chance for last words. There was one of my favorite questions. Thank you, Jim Pickett. There are obviously a lot of conversations that will continue. And selfishly, is there a plan for HIV Research for Prevention Conference now in late January, early February? Happy for you to answer if you'd like, or I'm happy to as well.
Well, I think what Mina said is certainly a commitment that we would like to work as rapidly as possible to present the primary findings in a peer-reviewed setting. And certainly we anticipate that that won't be the only story. As you've heard, there are multiple analyses that will need to be completed. So we hope that we'll be able to present kind of those data as they emerge at multiple conferences.
Great. I'll just say, putting on my R4P co-chair hat, between 083 and 084, there's a lot of important news to talk about, and we are working hard with the network and both trial teams to make sure that it gets full voice at R4P, and I suspect there'll be opportunities at the retrovirus meeting a few weeks later. There's a lot of things we need to talk about, lots of things into the future. I'm going to turn it back to Ntando in just a minute to close us out. But there was a great question, and again, apologies, I was going too quickly and I can't remember who asked it. about what else we have for long acting. As much as we're excited this week, the research and development process doesn't end. And wondering if all three of you might want to just for a moment think not only about what the future pipeline looks like, but the future of the research trials. There was a question that I got as well about what does this mean for standard of prevention in future trials? And just wonder, Yolanda, Sinead, and Mina, your thoughts about what comes next beyond cabotegravir and how we think about that. Go ahead, Sinead. It looks like you're ready.
Yeah, no, Mitchell, I think that's a great question. And I think we know that there are a number of interesting products in the pipeline and trials that are about to launch in the region in particular. We have heard of the monthly oral pill islatravir, which will be trialed. We know there's the Descovy trial and that there is interest in adding a capsid inhibitor to that trial as well, which is another long-acting product. So I think the pipeline is rich, and that's very exciting, but it does create a challenge for us trialists, because even with 084, as we saw, when you have two active products, you really are able to prevent HIV infection. I think there have been really interesting discussions that have been taking place in a number of settings, thinking about how we in fact are use alternative approaches to measure the efficacy of these products using external controls, either through cross-sectional incidence assays or counterfactual analyses, because we have become the victims of our own success in being able to do the traditional trials. And I think for all the trialists and people who've been involved in these trials, it certainly will mean a different way of doing business. So, it's exciting times ahead for all of us.
Yolanda or Mina?
I'll just echo that we have some hard work to be able to show new products are going to work well, but there is a nice pipeline, and I think that having more options is always important to the populations that need it. So we'll have to just figure out how to overcome our success.
Great. And Yolanda?
Yeah, I think the research pipeline is so rich, and what we have to push more is the products that are already out of the pipeline to ensure their availability, the quickest availability to those that need them most.
Well, you went on mute, Yolanda.
We are considering more on the CAB-LA. I think the issues of affordability will be very critical, and also to ensure that we also consider the issues of drug resistance and all that, something that is not coming out very clearly. But I think as advocates, we really want to understand in that aspect. But also, I would want to quickly point out the issues of gender inequalities, gender norms that affect women to access HIV prevention options. So these are most critical areas as we move forward with the products that are already out of the pipeline. Yeah, over.
That's great, Yolanda. Thank you, and it underscores we see this week a new option. So we have to go from developing these options to delivering these as choices. And thank you so much. I really want to thank all three of you for your insights. And I'm happy to turn this back over to my dear friend, colleague, and partner in many things, Ntando Yola. Ntando, thank you so much.
Thank you, Mitchell. Thank you very much. And again, our thanks go to our panelists, Sinead, Mina, and Yolanda. And we also thank the team that really brought this together, the communications team who made it possible to happen. We now have come to the end of this session. And I think, as many have said, as many speakers have said, it is indeed by no means an end of deliberations on what to do with this success. Because we now find ourselves in a better place with HIV prevention basket, having more tools to prevent HIV acquisition, especially for the communities that really need them the most. And I think it is important to highlight that the community teams worked on the study, named this study LIFE, and that stands for long-acting injectable for the epidemic. And I think it is within that vein of LIFE that we salute the over 3,000 participants who really worked hard with all the research teams and the research sites in the different countries who really helped to bring us to this scientific breakthrough. We are now about 10 years into the 2030 UNAIDS commitment to end AIDS as a public health threat. And this commitment is based on country-led movement to scale up HIV prevention programs as part of fast-tracking a comprehensive response to meet the global and national targets. I want to believe that HPTN 083 and 084 injects an opportunity to advance this commitment. And I think at that point, we'd really like to thank everybody. I think as Mitchell has mentioned, there will be a lot more conversations going forward to really figure out all of what this means. And thank you, everyone, and goodbye.